next up, we have Yaroslav Sych, or Sych, I don't know, from the University of Zurich, who's working with Fridtjof Helmchen, and he's going to talk to us about the mesoscale brain dynamics and how it reorganizes during learning of tactile texture discrimination. Looks very interesting, so take it out.
Yeah, thank you for hosting me here, and it's great follow-up for the previous two talks. Yeah, I'm Yaro, and I'll talk about mesoscale networks and how the connectivity changes during behavior. So our study mainly focused, or my interest was, how does behavior affect this global connectivity, and we use tactile texture discrimination task to assess it in mice. So we don't look only at the microcircuit at the baral cortex, but we also try to connect this whole basal ganglia thalamocortical loop, how the sensory information is fed into the motor circuits for mice to engage and complete the task. And we're also motivated by several recent studies, and not so recent as well, but there are just a few examples here, where people found the global brain activity with neuropixels probes, LFPs, multiple LFP probes, during behavior. We used multi-site or high-density fiber photometry for this. So we implant an array of thin optical fibers into the mouse brain. Here's the coronal section where we sample from basal ganglia, thalamus, cortex, and even hippocampus. And this part is implanted in the mouse longitudinally, and it's connected to the via fiber bundle to the wide field microscope. So on the camera, we can record a fluorescence image from the GCAM calcium sensor, which is a good proxy for the multi-unit activity in each brain region. Here's task which mice learn. It's the texture discrimination. So the timeline here is important. The texture is presented to the mouse whiskers. It's a head fixed task. At the second number 3, they have to whisk and discriminate between hit and correct, between go and no-go texture. And correct trials would be hit and correct projection, because for hit, mice have to lick and retrieve a reward, which is given at the second number 6. And for no-go texture, they have to emit any action. And of course, they can also do miss or false alarm trials, which would be error trials. Mice can learn this task very fast. Within a week, they become experts at the criterion of 70% correct trials for us. So here is the video example of expert mouse. So you see texture approach, and then for hit trial, mouse is licking, is retrieving a reward, and for correct rejection trial, it emits any action. What was surprising for me to see, here's the delta F over F of changes of calcium activity across 48 different brain regions in the mouse brain of the same hemisphere. Is the surprise for me was that the connectivity, the activation across these brain regions is so distributed and exists even in hippocampus and some of the midbrain nuclei. So, but because we look also during learning, we look at which behavioral parameters change. Indeed, we have also changed in some motor parameters. Mice whisk more and lick stronger, so they have a stronger vigor to retrieve their reward. And if we look at the changes in the correlation of these behavioral parameters to calcium signals, we see that the correlation to touch is a Pearson correlation, is increasing in basal ganglia, motor and somatosensory cortex, and ventromedial thalamus, while correlation to licking is decreasing. So the licking for a mouse is not the goal-directed motor command anymore, because they know that the texture which is predicting their reward. What we also see is that the discrimination power, if we simply train the linear discriminator on these calcium traces in this very early period of the texture presentation, from 3 to 3.5 seconds before mice lick on the first time on the water spout, we see that mice can discriminate the hit versus correct rejection trials. Calcium signals can discriminate in baral cortex, but also in basal ganglia, thalamus, and even hippocampus. This motivated us to look at the functional connectivity. We use the transfer entropy. It's basically a measure, a directed measure in our case, calcium time series, when the source area, the past calcium dynamics in the source area is predictive of the present calcium dynamics in the target area, given that the past of the target is not predictive of its own present. And if we just take the simplest measure of the this functional connectivity across learning is just simply number of links in this network of regions which we sampled from, we see that the number of connections is increasing throughout learning from the naive to the expert performance. If we plot the top 8 links in this network, so basically the subset of links which is increasing its connection strength throughout learning, which is shown here in this blue histogram on the left for heat trials versus the naive connection strength. We could see that throughout learning, mice established not only cortical-cortical communication between motors and somatosensory cortex, which has been previously shown and we would indeed expect it to happen, but also that the information is fed from the somatosensory baral cortex via thalamus back into the cortex. What was also surprising for us to see is that actually these functional networks during the time interval before the first link are very similar for heat and correct rejection trials. And we also looked at the network beyond the somatosensory regions, namely that we looked at the prefrontal regions and some of the posterior regions. We also could recapitulate results with the 12 fiber implants, which means that we have still increased functional connectivity throughout learning, particularly during the texture presentation time. And we also see globally that medial orbita frontal and prefrontal cortex became hub like regions because this early texture presentation period, cortical and subcortical regions were feeding information into these regions. And also internal globus pallidus, if we look at the changes in the clustering coefficient, also increase this clustering coefficient in comparison to all the other brain regions during this early texture presentation period. And clustering coefficient is basically the probability if you send outgoing links that your target regions which receive a link from you also establish a link no matter in which direction. So this is some sort of a hallmark of the pathways related like mesoscale computation for us. That's why we also related to clustering coefficient. You could also check details of this work in the bioarc. And here are the acknowledgements of the group of people I worked with, this Professor Fritjof Hentin, Alexei, and Mel. Thank you for your attention, and I'm open for questions.
Thank you for that's quite impressive, Yaroslav. There's quite a lot of stuff in there. I'm sure there are a lot of questions. I have one maybe to start. I'm also interested, I've been really interested in trying to figure out what changes during learning, but also what causes the changes that are bringing about the expert state. And I've been focusing on the idea that there should be some type of learning signal or lack of equilibrium that is present during learning, but that would be gone once you're in the expert state. You see? Do you see what I mean? So it's a slight, I think you've analyzed a lot your data from going to naive to expert, what changes. But in my view, there should be something in between that is present in between the two that vanishes once you're in expert because you finally, like you've reached expert state, so you don't have to learn anymore. Have you looked at your data this way?
Yes. So the beginning of this project was I was also interested in by this idea of Darwinian learning, right? That the neural circuit, you could speak about somatosensory, even like a microcircuit, that it would first during learning recruit multiple neurons of the similar tuning, be it auditory or visual, right? And then after the expert performance, it would drop out all those which have a tuning which is not relevant for this particular task. For example, it's the tuning for discrimination of 2 and 4 kilohertz, right? So in this case, the local circuit is further expanding in the number of neurons it recruits for making the task, and then it's collapsing, it's reducing again. And in my case, I was also curious about it, but first the analysis for this transfer entropy requires many trials because it does some sort of the bootstrap, because I end up with p-values for its transfer entropy, which is actually a link, not the magnitude of the transfer entropy itself. So my time step for the analysis is relatively coarse. It's only a day, right? I could take a day of the learning day itself. What I saw in some subset of mice is that indeed they first increased and some of them decreased their functional connectivity when they became overtrained, but that was only a trend. I cannot say it's like actually present in all mice.
Very interesting. Maybe to follow up on this, and since you mentioned, and maybe to echo Sean's talk, how did you actually compute? So you said you used a transfer entropy to determine the number of links, but I was a bit confused as to how you extracted the number of links using transfer entropy.
Not number of links. So for each region, I've calculated transfer entropy in the moving window, so the which maximize transfer entropy for the present time bin, and then the past 5 time bins were used for each region A and B, right? So A, B being the source and B being a target. So the source could transfer the information within this 5 past time steps to the target. And but basically 90% of links came from the last time step only, so the closest to the present time step of the target. And I did not extract the number of links, but if this influence is statistically significant, which is determined by the shuffling of this past time bin within the same brain region source A, the same type of trial, hit or correct rejection, but across the different trial, right? So once it's shuffled, you destroy this momentarily relationship between the time series, calcium time series, which you have. And then if it's shuffled and transfer entropy drops, then the p-value is below 1%, and that's the binary threshold for me of the effectively establishing the link. So it's a one link. And then I do all permutations for all possible combinations of brain regions, and then I get the adjacency or connectivity matrix that is still binary. And then I have 3 time samples, which is happening between the texture stops and mouse has a first lick. And from this binary matrix, I guess I get a weighted matrix of the connections, because some can be repeated, right? Basically, and some may not be in steps of 0.3.
That sounds good. Okay.
Yeah.
So basically, you count the number of inter areas that have this drop in p-value when you shuffle the data. Okay.
Yeah. So only significant ones I count as links. Yeah, which drop in the.
And then you scale them by the amplitude.
Yeah.
You had at the beginning, you had this plot you showed very quickly that you said this was the activity across the different regions, and it was flashing. It was a bit of a movie, right?
Yeah.
Right there. So, like, how come we're seeing the data in those circles? What's happening there? What do we see?
So these are multiple fibers. They are connected to the fiber implant, which is chronically implanted in the mouse. So the increases in fluorescence relative to the baseline are shown in the middle row, and the maximum when you see this white is about 5% delta 4F change. And this is the changes of calcium dynamics, which are related to the multi-unit activity of neurons sitting directly below the fiber tip. Each fiber is 120 micron in diameter. So from some of the Monte Carlo simulations of other people, the fluorescence signal drops by a factor of 100 on the diameter of the fiber down. So basically, I'm expecting a volume of 100 by 100 by 100 micron of the brain tissue contributing to this signal. So it's, if you want me to end up with estimate of number of neurons,
No.
That would be hard because you referred to Sean's talk. That still would be hard to guess, but I think it also depends on the brain region. I think it's in around 100.
Yeah. Okay. Cool. And I guess my last question, if there's not any, I don't see any other questions here. Is well, you've shown quite a lot of changes. Maybe and you've discussed a few summary statistics, and the hub stuff look really interesting. But did you find particular changes that you found were particularly interesting, or that were particularly striking your attention, that you were in the lab that you thought, wow, this is really surprising? Diving into that data.
Yeah, I think what was surprising for me is to see these connectivity matrices for 48 channel mice, and you can see that here is this red increase in the connectivity for prefrontal cingulate medial orbitofrontal cortex. Is kind of means that multiple brain regions are giving the inputs at this early period. It's kind of, you can think of it as the bottom up flow of the information. So that was impressive for me to see. I was even thinking first it's some sort of a bug.
Yeah, it is very interesting. And how did you create those hierarchical graphs?
These plots are basically the top 1 percentile of the strongest links from this matrix here, just to show the top strongest changes in the connectivity, which happen from the baseline period where the texture did not approach yet the whisker, and for the period of the this early texture discrimination period.
Oh, in a representation where the distance scales with the correlation.
In the representation.
I mean, we see that some centers are farther apart than others.
On this plot.
On this plot, right?
Yeah, it's a force-directed placement. Yeah, so it kind of tries to pull the nodes which have similar patterns, similarly projecting into each other, or supposedly similarly projecting to each other closer, while others further apart. Yeah.
Got it. All right. Well, with that, I think I'll close the stage. Say thank you to you again, Yaroslav, and to all the panelists. And well, thank you.